Okay. So I'd like to thank the organizers for the invitation to this great meeting. So we are the Cancer Regulatory Genomics Group, and we are part of the Department of Theoretical Bioinformatics of the German Cancer Research Center. As such, we're interested in, of course, transcriptional regulation, and in particular in the context of cancer genomes, transcriptional dysregulation. So we're interested in everything that goes wrong in transcriptional regulation. So there's no need to explain transcriptional regulation to this audience, but I'd like to make a distinction between two aspects, one that I would call genetic contribution, mainly sequence-driven binding of transcription factors of chromatin modifying proteins, and epigenetic contribution, which is driven by epigenetic marks such as DNA methylation or histone modification. So Hunter has made this distinction early on in a completely different context, but I will take over this distinction here. Now, this distinction becomes relevant when we look at what can go wrong. And in cancer genomes, we have, of course, a lot of sequence modification, in particular somatic mutation, single nucleotide variations. And of course, they might affect gene expression. And we're focusing on non-coding variants here in our cancer genomes. On the epigenetic side, we have extensive modeling of the DNA methylation patterns, hyper and hypomethylation, but also extensive changes in the histone modification patterns. And so I'd like to present preliminary results of two ongoing projects that would illustrate both of these aspects. So for the first aspect, we're focusing on the effect of single nucleotide variation on gene expression. And we have the chance to benefit from several large-scale data sets from the ICGC Cancer Genome Project. And in Heidelberg, we're involved into projects on lymphoma and child brain tumor medulloblastoma. So for these two data sets, we have whole genome sequencing, and so we have a large list of single nucleotide variants, and in particular non-coding single nucleotide variants. So we're looking at these data sets to try to understand how they might affect a gene expression. So if we look at these data sets of non-coding variants and we simply perform a functional analysis of these sets, we have like 400,000 variants in lymphoma, 200,000 in medulloblastoma. We see that for lymphoma, terms related to lymphoma or hematological malignancies are very much enriched, lymphoma or CLL, for example, here. And also for the medulloblastoma data set, terms related to blastoma, to various types of blastoma are enriched. So this seems to indicate that these mutations are not randomly put in the genome, but seem to target specific regions. And this is confirmed if we look at the recently published data sets of active enhancers across various tissues, and we ask, okay, which one of these data sets are most affected by these non-coding variants? And we see that for the lymphoma data set, enhancers active in lymph nodes are the most hit, the most affected regulatory elements. So these seems to indicate that there is some non-random distribution or selection of mutations in the non-coding genome. So we're looking for evidence of selective pressure on these mutations. And this is something that has been done already for coding mutations, and several methods have been published to find so-called driver mutation in coding genes based on the clustering of the mutation in the gene or the functional impact of these mutations on the gene function. And basically what we're trying to do is exactly the same based on non-coding mutation. And last year there was a nice tool approach that was published from the Gerstein lab called FUNSeq. It's an analysis pipeline for non-coding mutation trying to identify driver mutations. And so we applied this method to our data sets. I will show the results in a second. But we also developed an alternative complementary approach based on patterns of k-mers that are created or disrupted by these non-coding mutations. So basically what can happen is that a mutation might affect the binding site and hit an important nucleotide and disrupt the binding site. But of course, the reverse might also happen, that a mutation might indeed create a new binding site. And this is the example of the third promoter mutation that was largely shown in several cancer genomes. So what is very important in these kind of analysis is to have a realistic background model to which to compare what we're observing. And it's not a trivial task to do this kind of realistic background model, because mutations have a non-homogeneous distribution. They are focal regions of high mutations, and the mutation patterns are cancer-specific, as was shown by the TCGA consortium very nicely. And so a null model should respect these properties. And so we're using a local randomization procedure where around each SNV we shuffle the mutation locally in a window of various sizes, such as to preserve the distribution and the specific patterns of mutations. And having this background model, we can now compare what we observe. So for example, number of disruptions of binding site to the expected number. And we might have this situation where we have much more than expected, which would be a sign of a positive selection, or much less than expected, meaning that these binding sites are preserved, which would be a negative selection. So applying this approach to the lymphoma data set, we ran the FunSeq pipeline, and we identify a number of transcription factor binding sites that seem to be affected. So in green is the frequency of disruption of these transcription factor binding sites. In gray is the randomized, the expected number. And we see here a transcription factor for which we have a much higher number of disruption than expected, and the opposite here in this corner. And looking at the transcription factor here, they all, well, a number of them make a lot of sense, related to being expressed in blood cells or the lymphatic system. And we have, for example, here one prominent example is EGR1, which is a known regulator of tumor suppressor genes like P53 or P10. And so it makes sense somehow that this will be disrupted by the cancer cells, such as to shut down these cancer tumor suppressor genes. And here we have, for example, EBF1, which is a B lineage commitment factor, a known oncogene in a subtype of lymphoma. And so it makes sense that this one is rarely affected by these mutations. And this is a sign of a more negative selection. So to be able to do this kind of things, FunSeq relies on published transcription factor data sets from ENCODE, which is very nice, but it also limits the breadth of the study to the transcription factor that have been studied in ENCODE, and it also can only look at disruption of binding sites. So we thought we might develop an alternative approach based on simply k-mer creation or disruption patterns. So we're simply looking at which k-mers are disrupted or newly created by these single nucleotide variations. And we use our background model to compute a z-score of highly created, highly disrupted. And so we can have this scheme here of k-mers that would be frequently disrupted but rarely created, frequently created, rarely disrupted, or both of these cases here. And we call these k-mers oncomers because they might somehow be relevant for malignancies. So trying to interpret these cases, we expect to find more tumor suppressor motifs here in this corner, frequently disrupted, rarely created, oncogenic ones here. And here's a more complex situation where we have both creation and disruption of the same motifs. We believe that this might indicate rewiring of the transcriptional network here, and I'll show you some results. This is a heat map showing here each line is a k-mer being either highly disrupted and created, or specifically disrupted, specifically created here. So some examples here of our k-mers. So what we do is then we try to assemble these clusters of k-mers into longer motifs, and then we compare them to known transcription factor motifs. And here, for example, we find a motif that's very reminiscent of the GATA family binding site, and we found on the front-seq results we had GATA as a highly disrupted binding site, so it makes sense that we find it here highly disrupted. But what we can find here is a motif that very closely matches AML1 or RUNX1, which is a known oncogene in hematological malignancies. And so this one is frequently created and rarely disrupted. And so in this context of lymphoma, it makes sense. Here, a motif that might match the NFAT1 motif, which has a dual role, both as an oncogene and a tumor suppressor genes. Both of these roles have been shown, and so this might indicate that there's a rewiring, targeting of NFAT to a new set of target genes in this malignant context. So to illustrate the second part of the epigenetic contribution to gene regulation, we're focusing on DNA methylation patterns in a neuroblastoma data set where for 105 patients we have DNA methylation patterns. This is a collaboration with Frank Westermann from the DKFZ. And so what we're interested in is seeing the impact of DNA methylation on gene expression, both for promoter regions but also for distal regions. So if we look at the methylation patterns across all CpG sites, we have this well-known bimodal distribution of unmethylated CpG, a highly methylated CpG. And if we divide these CpG into three categories, the unmethylated, partially methylated, or fully methylated, we see that if we focus on enhancer regions or distal enhancer region, these intermediate category is very much enriched with respect to the whole genome distribution. And if we look at the variability of these methylations across our 105 patients, we can again divide into highly variable, stable, or intermediate. And again, these enhancer or distal enhancer are enriched in highly variable CpGs. So enhancer elements seem to be enriched in highly variable, intermediately methylated CpGs. And this is something that had been showed, for example, by the Schubler lab. Now, the impact on gene expression is pretty well known for promoter methylation. And we generally have a negative correlation between promoter methylation and gene expression. And if we look at the correlation distribution across all promoters in the genome, we have this blue distribution here. The red one is the randomized. And we see that we have an excess of negative correlation. Here is an example of a very nice negative correlation between methylation and expression, but we also have unexpected cases of positive correlation here. Now, we're more interested in what's happening at distal sites, at distal CpGs. And if we select distal CpGs that are more than 5 kb away from the TSS, then we consider any genes that are within 50 kb of the CpG we're looking at, and we're computing the correlation between the methylation and the gene expressions. And then we get this distribution here. Red is the observed and blue is the randomized distribution. And if we zoom at the tails of this distribution, we see an excess of positive, large excess of positive correlations and a slight excess of negative correlation between methylation and gene expression. And so this approach, by simply combining the CpG with any gene that is within a certain window, we can identify potential regulatory regions and their target gene if we have these highly positively or negatively correlated CpG gene pairs. So we're trying to understand what makes a difference between the positive correlation of methylation and the negative correlation of methylation. And if we look at ENCODE data sets, so luckily we have a neuroblastoma cell line in ENCODE. It's SKNSH. And if we overlap our positively correlating and negatively correlating CpGs with these data sets, we see that the negatively correlating CpGs are enriched in transcription factor binding sites, P300 marks of regulatory regions, whereas the positively correlating CpGs are very much enriched in K27 trimethylation here in this data set. And so there's a very clear distinction between the positively and negatively correlating CpGs. And this raises the question of the relation between DNA methylation and K27 trimethylation. And there is a different explanation in the literature, different models in the literature. Here's a model of a co-occurrence model where EZH2, which is part of the polycomb complex, recruits the DNA methyltransferases, and so we would have co-occurrence of K27 and DNA methylation. But we favor more an antagonistic model where methylation of the CpG prevents recruitment of the PRC2 complex, and K27 trimethylation is no longer set, and so the genes in this region might now be overexpressed. So to give you an example, this is a CpG here, which has a very strong negative correlation, not with the neighboring gene, but one gene further down. And we see here the correlation between methylation and the expression of this gene. There's absolutely no correlation with this gene here. And the nice thing is that this CpG co-occurs with P300 peaks in SK-NSH, which makes it a very nice candidate to be actually a regulatory region. If we look at the CpGs and the direct promoter of this gene, there's nothing. They're either completely unmethylated or completely methylated, but they do not explain changes in gene expression, whereas this one has this very nice correlation. So we have now many of these candidates, CpG target gene pairs, and we're starting to validate them experimentally using targeted assay to modify the methylation of the CpG and see whether the gene expression here is affected. So as a conclusion, for the first part, the non-coding SNVs show signs of positive and negative selection, and they might be the sign of an extensive rewiring of the regulatory networks of some of these dual genes, oncogenes and tumor suppressor genes. Now, we're currently looking at the effect on the target gene expression, and we're looking at the correlation with the expression of the transcription factor itself, because many of these transcription factors are differentially expressed, and so we're looking at whether this differential expression also correlate with changes in the binding sites. And we're extending this in a pan-cancer perspective as part of the pan-cancer project of ICGC and TCGA. Here's an example for lymphoma, medulloblastoma of k-mers that are both highly created in lymphoma, highly created in medulloblastoma. And so we're trying to make sense out of these pan-oncomers across different cancer entities. Now, for the DNA methylation, this correlation analysis allows to identify distal sites that affect gene expression, and this enhancer methylation seems to be very subtly modulating gene expression. The positive-negative correlation is depending on the chromatin context, which we try to understand better. Now, we're validating this experimentally using a CRISPR-Cas9 system. And we are looking more closely into this relation between histone modification and DNA methylation. And we're also looking at the relation between the SNVs and the methylation patterns, because if we look at these oncomers, the oncomers which contain CGs are much more frequently disrupted than other k-mers, and so this might be a link between SNV and methylation. So I'd like to thank the people in our group that did the work, Calvin, Sebastian, and Niels, members of the IELTS lab, part of the ICGC project, which provided the data set, and my collaborator, in particular, Frank Westerman from the DKFZ. Thank you for your attention.
k-mer to to produce the PWM matrix? Just to align the 6 k-mers and
so we have a procedure to assemble the k-mers, because very often we see that these are overlapping k-mers, and so we can assemble them into longer motifs and then convert them to position weight matrix like motifs, and then we can treat them as position weight matrices and compare them to known position weight matrices using different similarity matrix.
So assemble the k-mer like the de Broglie graph, or?
Oh no, it's a very simple alignment of words where we allow or not mismatches. Each k-mer has a weight on itself, which is a z-score, and so we can then turn this alignment into a position weight matrix. It's a very unsophisticated method.
Very nice talk. I had another quick question, also technical. The background model you presented, you mentioned that it's very critical to get that right. But we still don't know whether there may be other things that ought to have gone into the background model. Did you have any other controls to make, for instance, let's say the 1,000 genomes project and the distribution of variation, SNPs in that to see if the background model wasn't a too weak a null hypothesis?
Okay, yeah. So we have indeed some positive controls because, for example, in lymphoma, we have a phenomenon of hypermutation, somatic hypermutation that is well known, well described, where we know exactly which motif are targeted by these hypermutations. And so our positive control is whether, using our background model, we're able to identify these highly frequently mutated
Right,
motifs.
I'm more worried about the negative controls in that the background model is too weak a null hypothesis.
Oh, and too many false positives.
Right.
False negative. So, I mean, what we want to avoid are false
the
positives.
detection of TFs being hit by more SNVs, right?
So falsely identifying TFs that are.
Yes. I did a similar exercise with the 1,000 genomes projects at some point and found some of the same TFs being hit by polymorphisms in general more often than my background model would allow.
So we have indeed, when we compare the two data sets, as I just showed for the lymphoma, we find some of these TFs that are indeed affected in these two cancer entities, but for them there's good evidence that it makes sense that they're both involved. We have, on the other hand, examples, for example, JUN-D, which is highly affected in one cancer entity and and rarely affected in the other one. So showing that there's not a systematic trend to identify the same transcription factors across these different entities. Yeah.
Thank you.